ചിലവ് പരീക്ഷയ്ക്ക് ചോദിക്കാറുള്ള topic. So what is a phylogenetic tree? We have to start from that point. So basically, if molecular phylogenetics is the analysis of hereditary molecular difference, mainly DNA sequence, to gain information about organisms' evolutionary relationship. So there is a branch which is called molecular genetics. In this is molecular genetics, these people are mainly dealing with the DNA, because we have seen how a DNA sequence is present, and by simply analyzing the DNA sequence and comparing with the sequence of other organisms, scientists are now able to tell how much a relationship or how much these two species are related. So that branch is basically molecular phylogeny. And advantage of molecular phylogeny is that if you know one organism, and if we are able to take the DNA of that organism, from the DNA we will be able to get a DNA sequence, and you are comparing that DNA sequence with the sequence of thousands or millions of organisms in the earth. By simply doing a GenBank data analysis, you can easily tell which organisms are closely related with this particular DNA sequence or particular species. So that is the advantage of molecular phylogeny. And normally molecular phylogeny is expressed in the form of a phylogenetic tree. There is another word for a phylogenetic tree. It is also known by the name an evolutionary tree. So either we can use the phylogenetic tree or evolutionary tree. Now we will see how this is phylogenetic tree or relationship tree or evolutionary tree, and what are the features of this tree. Basically, every living organism contains DNA, RNA and protein. These are the molecular structures which are present in an organism. And there is one important point here, the second point. In general, closely related organisms have a high degree of similarity in their molecular structures. That means you take a chimpanzee and human beings. People are saying that nearly 98% of the DNA present in chimpanzee and that present in a human being or Homo sapiens are identical. So we have a close relationship with the chimpanzee, but we do not have that much closer relationship with other organisms like gorilla or orangutan or other kinds of it. So closer the DNA sequence, closer the DNA similarity, closer will be the evolutionary similarity between them. So closely related organism. For example, you take a lion and you take a leopard. Both are cat family, both are belonging to the cat family, and you are comparing the DNA of a lion and a leopard, and you are also comparing it with the DNA of a rabbit. You can see that the DNA of the lion and leopard are more closely related, because by simply looking at them, you can see they are more closely related. They both belong to the cat family. But that much similarity we cannot find in the case of a rabbit. So closely related organisms are normally expressing high degree of DNA similarity. Now molecules of organisms which are distantly related, as I told you in the case of rabbit and as in the case of lion, they are distantly related. So their DNA sequence is also showing more differences. So this is the one of the important rule. Closely related, closer the relationship between organisms, higher will be the DNA similarity. And those organisms which are distantly related may not be showing that much similarity, or their DNA dissimilarity or DNA difference will be higher. Now conserved sequence, we have to understand there is a term called conserved sequence, and these conserved sequences are normally present in mitochondria. You may have, while studying about cell biology, most probably you might have studied about there will be some small pieces of DNA in the mitochondria, and some small pieces of DNA are also present in the chloroplast. One important feature of this kind of DNA is that so in the case of this mitochondrial DNA or chloroplast DNA, they are conserved. The meaning of the word conserved is that they do not show any difference or change between the organism. That means if you take the mitochondrial DNA of a chimpanzee and mitochondrial DNA of Homo sapiens or human beings, we may not see so much difference between these. They show a high degree of uniqueness. They, without much change, they keep on going generations after generations. So that kind of DNA which do not express or do not show much difference are known by the name conserved, or there is another term, evolutionarily conserved. Conserved means they are remaining constant without any kind of change. So constant or conserved sequence such as mitochondrial DNA are expected to accumulate mutations over time. What is a mutation? Any kind of genetic change, especially sudden change. Any kind of genetic change where that happens suddenly are known as a mutation. You have studied about that. Now what happens is that this kind of mitochondrial DNA will be slowly accumulating mutation over a period of time. And these mutations will be there. But they are not expressing immediately because of the conserved nature of this DNA. Assuming a constant rate of mutation provides a molecular clock for dating divergence. Divergence means how two organisms are splitting into two separate, one organism is splitting into two separate species. That is known as a divergence. One species is gradually splitting into two or three different types of species. So by simply measuring how these mutations are occurring in the mitochondria, we will be able to predict that this organism might have existed during this period of time. Now evolutionary scientists and DNA scientists are able to predict with accuracy. If they get a fossil species, they will be telling this fossil species have been living on the surface of the earth around this much billion of years back. Because if they get the DNA, only DNA is sufficient. Based on that DNA sequence, they can make a prediction because they have data regarding other DNA sequences. So this is the advantage of studying about the conserved sequence. Then molecular phylogeny uses such data to build a relationship tree. Earlier I told you phylogenetic tree, evolutionary tree, and that same is known as a relationship tree. That shows the probable evolution of various organisms. We will come to that and how a this is a phylogenetic tree. So if you look at a phylogenetic tree, you can see that these two organisms are more closely related because they are closer to each other. Whereas these animals and these halophiles, they are more distantly related because one is coming from this branch, other is coming from another branch. So this is the advantage of looking at an evolutionary tree. By simply looking at an evolutionary tree, even for an ordinary person or layman, he or she can easily predict that these two organisms are very closer to each other. So we are keeping them very closer to each other. So that is the advantage of, so it is a very simple way of communicating how different organisms are related with one another. Now we will see what is a phylogenetic tree. So it's basically a tree like branching. Basically just like a different branches of a tree. It's a tree like branching showing the inferred. What is the meaning of the word inferred? Inferred means you are a guess. It's a guess. It's a prediction. We are guessing that these two organisms are closely related. So it's our guess showing the inferred evolutionary relationship among various biological species based upon the similarities and differences in their physical and genetic characteristics. So there are two, for example, you take lion, you take leopard, you take tiger. All these organisms are closely related. So if you look at their physical structure and if you examine their DNA, we can see that there is so much similarity between these organisms. That is why we are keeping them in a cat family. So by simply looking at this, we will be able to get an idea about the similarities and differences between this based on the physical and genetical characteristics. So based on that, we are placing this organism either closer to each other or far away from each other. So if we are keeping two groups closer to each other, that means that two groups of animals or organisms are having higher level of similarity at genetic level and also at the physical level. So that is the meaning of the word, that is how we are constructing a tree. So taxa joined together in a tree Implied means assumed or guessed to have a descendant. Descendant means formed from a common ancestor. That is the point they are mentioned. For example, here you can see the animals, fungi and plants. Here you can see that this is the entire tree of life. We are saying that these two organisms have a common origin at this point. It is after their common origin they have diverged into animals, fungi and plants. So that's how the evolution, so they are more closely related than the relationship with the other kinds of organisms like bacteria, or there are a lot of other kinds of organisms here, archaea. These are all more distantly related. So if two organisms are in the same branch, that shows they are more closely related. That is the meaning. So taxa joined together in the tree are implied to have descended from a common ancestor. Now we will see tips of the tree represent groups of descendant data, and nodes on the tree represent common ancestor. We will come to that. What is that? Here you can see that we are showing a small portion of a tree. That is, this is the entire tree. We are only taking this much a portion, and this is an enlarged diagram of that. Here we have shown three species are there. Taxon 1 is a species, taxon B is second species, taxon C is third species. Now what kind of information we are able to obtain from them? This taxon A and B are closely related because they have a common ancestor at this point. Both originate from the same ancestor. So we say that taxon A and taxon B have a common ancestor, or they have descended from the same node. So the tips of the tree represent group of different taxa, and nodes on the tree represent the common ancestor. There are 2 points here. One is a tip and other is a nodes. So this point is known as the node of it. Here there is a junction here which we call as a node. Node is representing the common ancestor, and this is the tip. This is the tip. Tip is representing the species formed from that, or descendant formed from that. So that is how tip and nodes are important. So tips of the tree are representing the descendant taxa, and nodes are representing the ancestor. Now two descendant that split from the same node are called sister groups. Here you can see that. Here you can see the taxon A and taxon B, your species 1 and species 2, have evolved or descended from the same node. So we call them as sister groups. Sister groups means they have more structural as well as genetic similarity. So because they have formed from or evolved from the same ancestor. So we are calling it as a sister group. Now what is the case in the ESO? Many in the tree below, A and B are sister group. They are each other's closest relatives. That is the meaning of the word sister group. And many phylogenies also include an out group. Then we have to understand a taxon outside the group of interest. For example, if you look at here, and here you can see the taxon A and taxon B we are considering as the sister groups. But here there is another species which is taxon C or species C. We call species C as a out group because they are not that much closely related. Because A and B have a common ancestor here. Taxon C had a common ancestor much before that, or more their common ancestor exists more older. So that is here there is a recent evolution. This evolution has taken place much more at an older time. That is the meaning of that. So they had a common ancestor at this node, but from that taxon A and taxon B have originated. But again here after some time again there is a further evolution forming taxon A and B. So that is why taxon A as well as taxon B are considered as sister groups, whereas taxon C are as a non-sister. We are not using the term non-sister or out group. So this is how we are calling it as an out group. Now a clade, there is another type. A clade is a group of organisms that include an ancestor and all the descendants of that ancestor. So that is another definition. So you can think the clade as a branch on the tree. Here you can see that a clade is a group of organisms that include ancestor and all the descendants of that ancestor. Here this is a clade. So this is one ancestor, and taxon A and taxon B are descendants formed from them. So we call this entire area as a clade. A clade means one common ancestor and two species or three species which have formed or evolved from that common ancestor. So common ancestor and the newly formed species are all part of the clade. So that is how we are defining a clade. So here the same thing is shown here. This is a clade. So this is a clade. This one, human, mouse, and a node is belonging to a clade. Here you can see that a node means a common ancestor, and human and mouse, because both are mammals. So that is why they are considering it as a clade. So that is how the definition of the term clade is given. Now what are the applications of the phylogenetic tree? It has a lot of importance or lot of applications. The main purpose of the phylogenetic tree is that if you look at this tree, you will be immediately able to tell that these organisms are closely related, these organisms are distantly related. That is the main purpose of any phylogenetic tree, to easily understand or predict the evolutionary history. Then they for example map pathogens and diversity for vaccines. For example, when coronavirus was discovered, very soon the scientists have told the entire world that this is a virus similar to the cold virus. How they discovered that? They have isolated the DNA and sequenced the DNA, and they checked the DNA and compared the DNA. Then they show that the DNA of the coronavirus is almost similar to the DNA of the cold causing virus. So they immediately understood many of the features of the virus. How we are able to treat the cold, or the way they are entering the body, is similar to how the cold virus are also entering the body. How cold virus are entering or infecting us, it would be then almost in the similar way, because they have a structural as well as genetic similarity. So by studying about the phylogenetic mechanism, we will be able to predict or map the pathogen or microorganisms, strains, diversity of that. Assist in the epidemiology of infectious disease. Epidemiology means how these organisms are spreading, how they are infecting, and how much time that will be taking to spread from one person to another person. These are all part of the epidemiology, how it is spreading in a society. Then aid in the prediction of functional novel genes. For example, you get a new gene because you get a new organism, and when we are examining that new organism with their DNA, we found that there is a new area in their DNA, and we found that it can be a possible new gene. How will you predict the function of the new gene? The best way is that you take the DNA sequence and you are putting it in the gen bank, and we will be doing a blast sequence that we have done in the lab. So that sequence of the gene will be compared with the thousands or millions or billions of other gene sequence, and GenBank will be immediately picking up most related genes. And if you get the most related genes, and if you are finding that they are expressing 99.5% similarity, then it is very easy for us. So if we know the function of the most closest gene, we can easily predict that this gene will be also having a same function. So that is how we are predicting the function of a new gene. So that is also possible as aid in the prediction of the function of the novel genes or new genes. In biodiversity studies, lot of applications are there. For example, recently I heard from television that there was an animal called dodo. You might have heard about that. And dodo became extinct in 17th century. I think the last species or specimen of the dodo was obtained around in 1653 or 54 something. Now scientists are trying to create a new dodo because they have the DNA. And based on the DNA preserved in the specimens in different museums of the world, scientists are now trying to recreate that dollar species. So maybe they may be successful, but they are attempting for that. But technically or theoretically we should be successful, but theoretically successful doesn't mean that practically we will be successful, because we are also working with the DNA. If the DNA is slightly, for example, my students are working on DNA. Sometimes what happens is that when we send the DNA sample for DNA analysis to Trivandrum at Rajiv Gandhi Center for, they said we are unable to get the DNA. The problem is that if any kind of contamination and is there, most of the time we will not be able to get the pure DNA. Another problem is that sometimes the specimen is preserved for a long time. If it is more than 1 month. Right now, if we are taking a specimen and you are sending it after 6 months, most of the time we will not be able to get the DNA. So how will be obtaining DNA of species which have been preserved around 300 400 years back? The preservation is highly good. The technique for extraction is highly accurate. Sometimes scientists may be successful. So it is not an easy process. Theoretically it is easy. Practically there may be a lot of challenges here. Then understanding the microbial ecology, how microorganisms, bacteria, protozoans and other kinds of microorganisms are performing. That also we can predict. Then inform multiple alignments. We will be coming to that. We have not studied about what is multiple alignment. Then identify the most conserved sequence in an organ. Conserved sequence simply means genes or sequence which remain constant without much variation. And those kind of genes normally present in mitochondria or chloroplast. And they are not undergoing much difference generations after generation. Their nature, their sequence are all remaining same after several generations. That is why we call it as conserved sequence. And this conserved sequence are playing a very important role in identifying a species. So this is regarding the applications of the phylogenetic tree. Now we have to see what are the different steps involved in a phylogenetic analysis. You can see that there are number of different steps. First one is we are selection of the organism or a gene family, choosing appropriate molecular markers. Why molecular markers means normally while genetics sequencing are there is CO1 gene. Maybe you studied in cell biology. CO1 gene is a gene present in mitochondria. Then there is another kind of gene, cysteinase gene. So this is also, so these are the two common genes we are trying. But there are also other kinds of genes, 70S or other type of genes are there. Some are also using that. So we have to identify one marker gene and we will be working on that. So amplification means we get only a small portion of DNA. Maybe 1 or 2 pieces of DNA. There is a technique called PCR. Using PCR, we are making thousands of copies of that. There is amplification, and from that we will be sequencing. Then we are assembling, and then we are comparing, and that process is known as alignment. Then, based on the alignment, we are preparing an evolutionary model. Okay, so this is how then we prepare an evolutionary model, and finally the reconstruction will be done, and based on the reconstruction we are evaluating the phylogenetic tree. So these are the different steps which are involved in phylogenetic reconstruction. Then so there are number of different types of methods which we are using in phylogenetic reconstruction, and the assembly and align data. The first step is to identify a protein or DNA sequence of interest. Assemble the data set consists of other related sequence. And what we are doing for finding out is that most of the time we have to identify either a protein sequence or a DNA sequence. That is the first step. And then what we do is that we will be taking that sequence most of the time by doing it from NCBI blast. We are running an NCBI blast, and we based on that we will be getting some of the most closer sequence, and these closer sequence are also taken. Then we will be doing a comparison, and that comparison is normally done by a process known as multiple sequence alignment. There are number of software for that: Cluster W, MSA, MAP, T Coffee. These are also all software used for gene comparison. That is, we have 3-4 genes and we have to compare them, how they are closely related. And for that we are using some software, and these softwares are Cluster W, MSA, MAP, T Coffee. And what is the function they are doing? They are performing a molecular sequence alignment. We will study about that also. Then second process is that build phylogenetic tree from sequences using computational or stochastic model. Stochastic model basically means based on computer inputs, the computer will be able to make a, for example, next class I will show you how this kind of modeling is working, because in the next class we are going to take a DNA sequence or a protein sequence, and that only we will have protein sequence. When that protein sequence data is put into a software called RAS mode, it will be showing a 3-dimensional model. So this kind of model preparation can be done presently using software. So to build a phylogenetic tree, statistical methods are applied to determine the tree topology. How the branching of the tree should be there. That is the meaning of the topology. Then calculate the branch length. How much, for example, this is the branch length. How much the length of this branch? How much further this branch may be from the common origin? This branch has a longer branch length. So those kind of points has to be estimated. Then most common computational methods applied are distant matrix method, discrete data method, and maximum, such as maximum parsimony and maximum likelihood. These two are a completely different topic. We will be studying that. Then there are a lot of software for this preparing evolutionary tree. Just remember that these software which are used for preparing evolutionary tree are POP, PAML, file. So sometimes there may be questions. Give an account of, give examples for some softwares which are used for preparation of evolutionary tree. There are POUP, PAML, and file. These are the most common type of software used for preparing phylogenetic tree. And finally, we have prepared a tree. Then we have to assess the tree, whether this tree is correct, whether this tree is biologically sustainable, or this tree will be accurate. And that process is known as statistical testing. And that processes will be also done. And tree estimating algorithm generate one or more optimal trees. So there is a software for that, and that software will show you 2-3 trees. Out of the 3 or 4 trees, we have to select only one tree which has more probability, more chance of being real. And we are analyzing that, or we are selecting that best tree based on certain statistical methods. So that is a beginning. And that is also, there are also software for that. For example, bootstrap, jackknife, reassembling method. These are some of the softwares which we are using for analyzing which is the best tree based on statistical probability. Then bioinformatic tools for phylogenetic analysis. Here you can find there are also plenty of software. This is a completely very detailed, complicated, because branch, maybe if you want after your degree, you can go for bioinformatics. MSc in bioinformatics are there. MSc in evolutionary biology are there. And if you, even in India, for example, last year there was one student. Now I think he went to Trivandrum. In Trivandrum there is a university called Digital University. You might have heard about it. There they have this kind of program, ecological informatics. So if you go for that, those kind of program, it may be initially it may be difficult, But still, then the the student who was studying here, who has went to this digital university, he is studying bio AI. Bio AI. You know what AI means? Artificial intelligence. And this bio AI means, because why this AI, artificial intelligence, is so important? Because most of the business billionaires are investing a lot of money on artificial intelligence. Whether because in the next generation, artificial intelligence will be determining the life of the people. That is why they are investing so much money on that. Yeah, recently you might have heard about a software called Chat GPT. Have you heard about that? Chat GPT? Chat. Now if you just check in your Google Chat GPT, and you can log in into that Chat GPT using your Google account, Google Mail, and try and ask that Chat GPT. It's actually a robot. You ask any question to Chat GPT, just like Google will be bringing you answer, this Chat GPT will be giving you an answer more like a human. It may be short answer. Any question you can ask. So that's how the things are going. Now, so here also you can find a lot of software which we are using for phylogenetic analysis, Panther, Peapod, Pfarm, Treeform, Phylofile. So thousands of softwares are there, and this is a highly emerging field. Every day new softwares are introduced. So these are basically two types of most common method. One is distance based method, and out of the distance based method, UPGMA. This is one common method which we are using for phylogenetic study. There is another method called neighbor joining method. Both are complicated method. Now I am not going into that. Then here there is a technique called maximum parsimony method. According to this maximum parsimony, this states that simple hypotheses are always preferred to more complex hypotheses. And this is also known as principle of parsimony. Parsimony simply means in Malayalam we can call pishik. That is small. The basic idea is that phylogenetic tree with the fewer evolutionary steps are always preferred by nature. That is, you are preparing a tree with a lot of branches, and you are also preparing another tree with fewer branches. The nature will be always preferring a tree with fewer branches. And that principle is under principle of parsimony. Nature is always preferring phylogenetic tree with fewer branches are there, or fewer evolutionary steps are always preferred. So this is how the phylogenetic tree are prepared. Here also you can find a lot of details regarding that. I am not going into that right now. This is the software used for preparation of the phylogenetic tree. So today you are closing here. And in another class we will take some more details regarding the phylogenetic.